I'll go ahead and kick off the main part of our program. I'm going to use a few notes. I just want to welcome everyone to, it's really exciting for the Grange to host this series virtually. We really welcome all, and that includes many colleagues and neighbors and newcomers. The attendance at our virtual series has really grown. So thanks. If this is your first time at the Grange, we would normally recognize you in person, but we're recognizing you virtually right now this time. I am Elizabeth Lee. I'm the Director of Education and Interpretation at the Lake Champlain Maritime Museum and a Grange board member. Tonight I am going to be, you're going to see Andrea Barrett in the background. Andrea is another Grange board member and Lyceum committee member, and she'll help me out with some of the background management. And for those of you who this is your first time attending a Lyceum series, the Grange is a really amazing community, which involves lots of events and activities, a lot of volunteers, and we have, with some great support from the Glenn and Carol Pearsall Foundation, we're able to change our in-person Lyceum series into this online series. The Lyceum series itself is over 10 years old and just keeps getting richer. Many of the programs are available online, and specifically last fall's Lyceum series is available on YouTube, so you can check that out through the Grange's website. The Grange is supported by donations. We're happy for you to donate. Again, you can do that through the Grange website, and it helps us just to keep doing what we're doing. I also really want to thank an amazing cadre of volunteers, from the board members to the people who work events, the in-person events, to those who have contributed to the Lyceum series and lots of the other upcoming events. So thank you so much. A really great group. With that, I think I'll go ahead and get started with our program. So tonight we are going to hear a really interesting topic about mycology. This was our kind of keynote idea. This year has been so sort of strange and mixed up, and things have seemed so entangled. It seemed like a great theme for this series, and to thread together some really interesting ideas. Our guest tonight and speaker is Susan Hopkins, and she is an incredible mycologist who I met years ago in the field collecting and identifying mushrooms, and also have heard her present at the Adirondack Research Consortium conference. So you're in for a treat. I will turn it over now. I will, and actually I'll just mention also, don't forget, we will see you once a week for the next couple of weeks for some of these other very interesting and entangled stories. Okay, so I will let you take it away. Susan, you can go ahead and share your screen and feel free to introduce yourself. And I'm gonna turn off my video while you're speaking, just so you won't, the audience.
Okay. Can you
actually, one really exciting comment tonight. We have folks registered from Tokyo to San Diego and all over the Champlain Valley. So welcome everyone, international, national. We are going global. We are totally going global. Welcome.
Okay, so you can see my screen, the first slide.
Yeah, you're all set. Go ahead, Susan.
Okay, I'm going to do a slideshow to make it bigger, I hope. Okay, and everybody can hear me all right, correct?
Yes, that sounds great.
Okay, so we're talking about how fungi work in our world. And I have a beautiful picture here of mushrooms that I collected from Elk Lake last year in late August. And when we see the great variety of what we have here, it's really just the tip of the iceberg of what is out there. And these are also the fungi that we can see, which is mainly, I'm going to talk about how the fungi we see grow. And over here is a cartoon from Sweden that sort of shows what we're going to be talking about, how the mushrooms are very connected to the trees and plants and what they're exchanging back and forth. You can see that without even reading Swedish, but sugars, water, nitrogen, I think, and phosphorus. But anyway, we're going to talk about that a little bit more in depth. I just wanted to talk a little bit about my own experience. I moved up here about 12 years ago from New Jersey, where I was a member for a lot of years in the New Jersey Mycological Association. This is a year-round club that does all sorts of activities, and main purpose being education. And we have public events where we once a year have a fungus fest at a large auditorium. But the main learning experience is here. We go out in the woods for an hour or two and then come back, set things out on a table and look at books and try and help each other from experience. We are also lucky enough to have some very good speakers. If anybody might recognize Gary Linkoff right here from, from the New York Mycological Society, who has sadly passed away. He's also the author of the Audubon Society Field Guide to Mushrooms. I will say one thing, I am not a professional mycologist. I'm an advanced amateur, having done this now for actually 41 years. So my explaining a lot of this, here we have the entire kingdom of fungi as it was a couple of years ago when this book was written. And we are only going to talk about a little bit of a group here, a few over here, and this group down here, which are mostly the crop related fungi. So what is a fungus? This was the simplest explanation I could find. And the main thing is that they're multicellular. Their cells have nuclei. They are heterotrophic, which means that they can't make their own food. So they have to get it from somewhere else. They absorb their food instead of ingesting it, which I'm going to explain each of these a little bit more. And they are develop this rather diffuse branch tubular network. What that means is this kind of spiderwebby thing right here. And here, this is colored that wood because of the hyphae, which are the next thing I'm going to show you. And then they reproduce by spores, which are sort of like seeds. So here is the hyphae. This is what the individual real body, vegetative part, whatever you want to call it. This is the fungus, these strands. Usually you cannot see them. They're so microscopic unless they get into a mass, mycelium, or in the case of one mushroom, and probably some others, they form very distinct strands that you can see. But what you need to know here is that the fungus puts out enzymes at the tip to digest whatever the food or substrate is that it wants. And then it absorbs back the decomposed organic matter back into the hyphae and sends it down the network. Now here we're able to see the hyphae a little bit more en masse. In the case here, it was from a tissue culture we did at Rutgers many, many years ago. The mycelium radiating out from the center, trying to find food, or in this case, feeding on the potato auger that we have put it on. And what's interesting to me is that the chicken mushroom is a yellow mushroom and it's yellow. The bluet is a kind of a lavender purpley thing, and the mycelium is purpley. And then the pink oyster is also pink, as it shows here. So let's look at the life cycle just for a few minutes. We've looked at the hyphae a little bit, and two hyphae have to come together. Each one of the spores, as they develop, has half of the chromosomes material that are needed to form a continuous ongoing mycelium. But two strands have to come together before, if conditions are right with moisture, temperature, food source, before they can form a mushroom, which the only purpose of this fruiting body, the mushroom, is for reproduction. It's like, you have to think of it sort of like the apple on the tree. Its only purpose is to to create the seeds for reproduction. So there's a whole lot more going on underground or wherever it's feeding. Like here, this is just a handful of soil that we dug up a couple years ago. And all of this white business is some kind of hyphae or mycelium of some fungus, and this gold would be also. So spores, the microscopic seeds of the fungi. This is in my kitchen a couple of years ago, but they're usually fairly easy out of the fruiting bodies again that we can see. There's a whole lot of other ways fungi can make spores or reproduce somewhat, but for our purposes, the fungi we can see, you just put it down on a paper, maybe cover it with a cup, leave it for a few hours, and you'll get some sort of spore print. Now this helps us identify often which group we're looking at, sometimes by the color, sometimes by the shape of the spore, or where it has formed the spores is the next important, where it's formed its spores. Now there's two major groups of how the spores are formed in the fruiting body. The one is ascomycota or sac fungi. They're just little long sausage-like cells. The hyphae grow into these long sausage-like cells, and then there's usually 8 spores formed, and then it will open up at the top when they're mature. If you notice here that half are one color and half are another, that's what sort of divides the material that's needed for it to reproduce, so that when it meets in the ground or the substrate, the 2 strands come together and then they complete both parts of the chromosomes, as it were. So we have these cup fungi are a big major group, and all of these sacs line the inside of the cups. And some of you might know about morels or have eaten morels, and all of these little pits here are lined with these sacs for the spores, with the spores. Now, the other major group that we're going to talk about, which is probably more of the fungi that we can see, even though the ascomycetes are a bigger group worldwide and more species than the basidiomyces, but these form, their little hyphae form into these little club-like structures that have little points on them that turn into little spores, again with half of the chromosome material in each. This is an unbelievably good picture of the basidia on the face of the gill. Sorry. If we looked at the face of the gill right here and squashed it on a mount, this is what we would see. Although in this case, many different mushrooms have many different spores and shapes and colors of the spores, but also there may be some other sterile kind of cells in between here that are also important for identification. I am personally one that tries to avoid using a microscope, but sometimes you have to if you really want to know what it is. So where are the places that the spores form? We already said the cups, the long cells with the spores in them, as in cup fungi. And then the mushroom may form gills, may have spines or teeth, like here. And it may be pores in a fleshy fungus. It may be pores in a woody kind of tough fungus, or it may be some kind of coral or spindly thing like this that just forms the spores, all the cells that make the spores all over the outer surface of the mushroom. Okay, now the next important thing to look into is how do they grow? How do they feed? We said mushrooms can't make their own food, so they have to find it somewhere else. And they do that in one of 3 ways. They can either be a saprotroph, a parasite, or mycorrhizal, which is the one I think everybody's interested in now. Now I want to say something here about Latin names and common names. I've put the Latin here, but you can get into trouble with common names because they are not always consistent from book to book. Or if you really want to know something about this little parasite on this russula, you're going to have to use that name. You might be able to Google parasite on a russula and come up with this, but not necessarily, or it might not be the same thing. So you need to sort of be aware of that when you're using some of the common names. For this Boletus edulis here, I can think of at least 4 different common names for this. Sepp, Porcini, Karl Johann if you're in Scandinavia, Steinpilz in Germany. So there's quite a lot of variation in the common names. And also different field guides and books will use different common names, and it may also be regional common names. So just be aware of that. So the first way we're going to look at how they feed is the saprotroph, which is what the mushroom is feeding on. That mycelium that we looked at, the hyphae, is feeding on dead organic matter, and they are recycling mainly carbon. Mushrooms in general are recyclers. Since they don't make their own food, they have to break down something else. So they are a great garbage. As a friend of mine used to say, trash burners without smoke. So now all 3 of these are good edibles if you are sure of the identification, but there's many puffballs. But there's several species here that might be called lion's mane. There's also several species that might be called oyster mushrooms. And these 2 are easily cultivated. So I would say that's the way you really want to go. Now, we should talk about here in the Adirondacks, the white rot and brown rot. What happens here is you can see the mycelium of the fungi working on a log that's already dead and breaking it down into a very crumbly, waffly, soft material. And it's very common. A lot of fungi here in the Adirondacks are white rotters. And you can see this, well, maybe not with the snow today, but you can see it often in the woods. Now, brown rot fungi are also pretty easy to see, and they tend to eat the cellulose out first, whereas the white rot eats the lignin. It's one of the few things, fungi are one of the few things that can break down lignin in wood. And then here, the lignin is left in this cubical form, whereas the cellulose has been already eaten out. What both are doing is turning the organic matter back into a component that can be used by some other organism. So if we really need fungi to do this all the time. Now I love this picture because there's 2 vastly different species on this same old dead log. And in the case here, the horsehoof fungus, tinder fungus is another name. It's a white rot working on this dead conifer stump. I think it's Norway spruce. And then this other red-belted polypore, Fomitopsis panicola, is a brown rot. The only reason they can exist on this log together is because they are eating 2 different parts of the wood. This one's eating the cellulose, eating the lignin, whereas this one is eating the cellulose. I have to say that stump is no longer there. It's totally decimated. Other important rotting fungi here in the Adirondacks, Tricaptum biforme, this turkey tail. This is a false turkey tail. Usually when it's young, it has this kind of lavender growing edge. The true turkey tail is the Trametes versicolor, which is a little bit more semicircular, brown often. Often used in medicinal extracts for immune system enhancing. And then the chicken mushroom I don't see that too often because it grows on hardwoods. If you find this is a species complex. That means there's several species that are closely related to each other. And the main thing to know is if you're interested in this, don't eat it if you find it on hemlock. There's something that is not going to be good for your system. And there have been poisoning cases reported from that if it's growing on conifer, specifically hemlock. Okay, so then we should talk a second way that mushroom really feed is either as a parasite or a pathogen. This is probably the biggest parasite pathogen here in the Adirondacks, in the honey mushroom or Armillaria mellia. This is another complex. There are at least 7 or 8 different species in this complex. I can honestly say I've seen 3 different ones here in the Adirondacks, but they're very close to each other and very similar. So I think you'd really need a microscope to tell them apart. But you can see their mycelium or rhizomes here, rhizomorphs. If you peel back the bark, very often in a lot of trees, you'll see these kind of shoelace looking stuff, which is the vegetative part of the honey mushroom. This can cause a lot of timber damage. But why doesn't it kill everything? Well, there are other fungi that work in the opposite direction to prevent it from killing every tree in the woods. Now, a couple more parasitic fungi here in the Adirondacks. This is one of my favorites, Phaeola schweinitziae, the dyer's polypore, because I use it to dye wool. It's very good source of dye pigments for me. It makes a wide range of colors, but it's also a pretty specific rotter or attacker of conifer roots. And sometimes it will take a long time to really kill the tree. So it can be there for a long, long time, but it also will continue to grow come up every year about the same time after the tree has died. Now, chaga is one of the hot topics here. Inonotus obliquus is the actual name. I've only ever seen this on standing up birch trees. And this is, it's not actually a fruiting body, but it is the fungi that have distorted the bark into this funny looking horn-like storage body. It's a conch kind of a thing. The actual fungus fruiting body is this was taken about 10 years ago. And when it finally died and I pulled it off, the tree totally was coming down. The pores were revealed just below where the fruiting or where the conch was. And here are the pores, which is why it is still put in polypores. Very interesting fungus, but also used medicinally for immune system enhancer. Making a tea, it's like concrete to deal with, so you have to sort of break it up and steep it as a tea. Now, just some other odd, interesting fungi. The only one that really grows here in the Adirondacks that I know of is the lobster mushroom. It's parasitic. It's a fungus on a fungus. It's this red ascomycete, the long sausage cells on a normal basidio mushroom, a lactarius or a russula, that would have had gills, but the ascomycete, the lobster, the hypomyces rather, has distorted the form into this funny looking, beautiful lobster colored thing. It's a good dyer too, but the only part I want is the outside edge. Now, corn smut is considered a delicacy in a lot of Central America and Mexico. If you have corn, you're probably familiar with this. I don't know how you eradicate it or how you avoid it, but it is kind of it's kind of very noticeable when you see it. You'll first see these very tight little clumps. Now, cedar apple rust, this is getting into a lot more complicated kind of a life cycle than I can explain, but it has two hosts at different stages in its life. It will grow on the cedar tree as we have here. And when it's raining and very, very wet, it sort of exudes this orange-like spaghetti. And the spores form all on the outer surface of this. If you wait a few days, it'll turn into a gooey mass with orange, dark, like dirt or dust or slime all over your hand, which is the spores. And the other cycle is on the apple tree, which I honestly can't say what that looks like, but it's a definite problem for growing apple trees. Now this I just had to throw in because it's just an unbelievable picture with this ant growing with two little mushrooms right out of the top of it. But parasitic fungi on insects is quite common in all different kinds of insects. This particular one is tropical. Okay, now the real subject of our talk, I think, is our mycorrhizal or mutualistic symbiotic relationship, association of fungi with plant roots. And here I've listed some of the benefits of the mycorrhizal associations. can increase absorption for the tree or plant, can explore greater volumes of soil. This is something that's a little beyond me, except that I'm sure the chemistry is important for the actual exchange of the nutrients. Beneficial for plant partners in nutrient poor soils, deserts, sandy soils, poor nutrition. What they're exchanging is nitrogen, phosphorus, and potassium in exchange for the products of photosynthesis, which is the carbohydrates or sugars. One thing a lot of people don't realize is the mushroom partners will make the trees and plants a lot more disease and drought resistant. That is, they can fight off, if a tree or a plant has multiple, usually, mushroom partners, they're much more able to stave off invasion of some other organism, whether it's insects or other fungi. And also this is kind of astounding that 95% of plant families form some type of mycorrhiza. Mycorrhiza, literally fungus root. Now, there's two basic kinds that I'm going to talk about because they're the ones I sort of understand best, but right now I think they've decided there's about 7 different kinds. Although I have to say, when you get into it, there are a lot of strange combinations of fungi with plants and trees. But one that's very well established and known is here, where the hyphae of the fungus enters the individual cell. There's the cell wall of the plant or tree and forms these little branch-like clusters, and that's where the nutrients exchange, the plant providing the sugars, the fungi providing the nitrogen, phosphorus, water if it needs it. Example here of maple trees, you're not going to find a fungus that you can see under a maple tree unless there's other ectomycorrhizal fungi trees nearby, because these guys don't form fruiting bodies that you can see. They're all microscopic, so they're not as interesting to me because I don't see them. But they are important for crops. Most crops, these are just a few examples here, have mushroom partners that are important to their health and growing. My friend who helped me with this, she says, golf courses, if they over-fertilize their golf course, their grass isn't going to grow because they're deterring their mycorrhizal partners by too much fertilizer. But I have to say, this is not an area that I know a lot about, except that these are important things that we want to continue to have and growing well. So the one that's a little more commonly understood, but less of a factor in the world as a whole, is the ectomycorrhizal. This is where the fungus wraps around the root sheath and then goes between the cells here to exchange the nutrients. And these pictures are pretty astounding, microscopically somewhat. I haven't tried this, but I want to pull up a root and see if I can see this. But this produces the mushrooms that we are used to seeing. Now this is pretty small up here, but if you can read it, the nutrients can flow through this network of mycelium. Here's the roots of the tree, and here's the mycelium of the fungus. But they can also connect many different species of trees together, and also to cross species with the nutrients and whatever is needed by one and the other. And the other thing is that if there's something attacking one of the trees, it can send some kind of, it's either chemical or electrical or whatever. That's getting a lot of study now. How is it that they actually communicate and say, in this case, the sugars from an older tree to end up in the shaded seedlings, and minerals to cross from one fungal species to another? I mean, that's pretty amazing. this is an area that's getting an awful lot of research lately, and it's pretty astounding. I have a list of some further reading at the end that it's amazing what people are learning, mycologists. Now, this is a species that we have here in the Adirondacks, or a very close variation of it. And destroying angel is a fairly well-known, but also cosmopolitan name for this. All white, all deadly poisonous, as it sort of indicates in the common name. But ours here may be slightly different from the one in Europe, but it's turning out it's pretty much the same. Now, this is another slide to sort of illustrate how important the mycorrhizal is to the little tree. This tree might be, what, maybe 6 inches high. Its roots goes down maybe this far into the ground. And then the mushroom, mycelium, hyphae, can go further down into the ground to absorb all kind of nutrients. But what he's saying here, colonize multiple plants forming a common mycorrhizal network. It's like the internet. This is the internet of the natural kingdom. This is where all kinds of communication, exchanges, the forest as a whole needs all of these networks. And the other thing that is amazing to me, which I've learned only in the last couple of years, is that they can be formed with numerous plant species for these ectomycorrhizal fungi. And I just want to say, because I saw this slide, Swillus bovinus, I have seen this mushroom in Sweden. And then 2 weeks later, I saw it here in the Adirondacks, and I said, wait a minute. Well, this is mycorrhizal with Scotch pine, or Pinus sylvestris. And I thought so I finally got that confirmed, that this mushroom came in on the roots of the seedlings from Europe when it was planted here in the Adirondacks. I've only seen it in 2 places side by side here in the Adirondacks, but it's come in on the roots of the seedlings that were planted about not quite 100 years ago. Okay, so some genera here in the Adirondacks, or I like to call this the big 5+1. They are all ectomycorrhizal. That is, the root system of the fungus connects to the tree roots and forms the sheath, and it goes in between the cells of the root to exchange the nutrients. So Amanita, bolete, boletus, cortinarius, lactarius, russula. And I put chanterelles in here because it's a good picture, but also this is a highly sought after edible. The point being that if you have a place for these, since they are mycorrhizal, they will always come up in the same place. And so year after year, usually about the same time of year, although I have seen this flush in this particular spot where that picture was taken 3 times in one summer. Depending on the weather, they like a lot of water. The other thing is this mushroom being highly edible, it can't be cultivated like the shiitake or the lion's mane, because it has to have the tree association. So to know where it grows is a good thing. Now, I just want to talk a little bit about a study I was involved with, but also the people that are really studying the subject of this interconnected network, this internet, as I have called it, but the communication between the trees and the fungi. This man right here is Ritas Figalis, who is the head of mycology at Duke University. And I've run into him a couple times, but I've collected these mushrooms for him. He's particularly studying this genus Swillus, which is a subgenus of boletus, that is fleshy with tubes or pores as their spore-bearing surface. The spores form in these tubes here, and they come out the little holes. But anyway, Ritas has been studying this group for quite a number of years, and he wanted spores of these species so that he could inoculate seedlings of white pines to be able to, and don't ask me what the thing was that they put some sort of marker so they could see where one thing was moving from the tree to the fungus or vice versa. they're still working on it. I know. Here are his two graduate students, and this was a mushroom conference that I was at. And these were mushrooms that I brought for him, including right here and right here, for them to take tissue culture samples right when I got there. So you can see Ritas is very happy. And these are all, by the way, these are all mycorrhizal specifically with white pine. White pine will support a lot of different fungi, but if you have Swillus pictus, like Gary used to say, or Spraguei now, you have white pine. It's there somewhere, maybe 50 to 100 feet away, but it only grows with the white pine. Now, an interesting thing here about this double name, in all of your current field guides, it's going to be pictus, but the Spraguei, I think, is an older name. I'm not sure. I don't think it's European. I think it's just earlier. And according to the rules of nomenclature, the earlier name has to apply. Now, if we want to talk about some really strange relationships, every time I'm on a mushroom walk, say, oh, look at that, the Indian pipes, are they a fungus? Well, they're not a fungus. They're a non-chlorophyll plant. They cannot make their own food. But what has been discovered is that the trees, I'm not sure which one, or it may be multiple species, the trees provide the sugars, carbohydrates from the photosynthesis, but it's the Russula, genus Russula, not necessarily this one species, but the genus Russula, which is very common, provides the internet or the network for the sugars to go through the fungus down to the Indian pipe, the Monotropa uniflora. And so it doesn't need any way of making its own food if it can steal it from somewhere else. The work that these two do, this one benefits from. So it is considered a parasite on the other two. But for a long time, people thought it was just one way, but it's one way through the fungi. Now, I just briefly talk a little bit about truffles as a group, because everybody's interested in them, that they grow below ground. They've evolved to be edible to some animal or somebody, not necessarily humans, although these two are very choice edibles. And they have developed an odor, emit an odor when the spores mature. So a chemical signal to the animals that the feast awaits, which is important for the animal to eat it, so they carry the spores somewhere else and poop it out, and that gives it a little nutrient packet to start to grow. It's a fascinating relationship that has evolved over millennium. But here in the Adirondacks, our first truffle or truffle-like fungus that we found was a couple of years ago. Now, Mike Castellano is a world truffle expert. He's also a Paul Smith graduate, but he went to Oregon State to learn the next step in truffles. But he was here, and 5 minutes after he drove away, we found this truffle. But we do have this species as common here. I believe it's on Laphomyces, but you will only see from arrow up above the ground, and you have to be very careful and excavate the yellow mycelial cord down to the truffle-like fungus below the ground. Very common in the Adirondacks here. An interesting fungus, parasite on a mycorrhizal fungus with probably, well, I'm not sure, but probably again, the pine trees, the Laphomyces. I'd have to ask Mike about that. So if we look at the forest as a whole, this is a fairly crude, simple drawing, but I like it because it's easy to understand in that they've color-coded the water and the sugars going from the leaves down to the roots of the fungus, and sort of noting some of the other little things that are happening. It's the community of the forest as a whole that's important here. And my friend also pointed out that there's a lot we don't know in these relationships. This plant over here Yeah, it's got leaves. It can photosynthesize its own sugars and carbohydrates, but it also can get its food from a fungi network below ground. Mycoheterotroph, that's what it's called, where it has the association of the two places that it can get its food from. And well, so there's a lot more complicated sorts of relationships that I'm afraid I'm not the person to help you understand those. I've just tried to give you some of the basic forms that I know about. But we also see a saprotrophic, so we know this is something that's going to feed on something that's already dead, a fungus feeding on dead organic matter. We have the mycorrhizal relationship here of ecto with the amanita connecting to the tree roots. And then they show what looks like a bow lead over here. All of the bow leads are connected to the roots. They are all mycorrhizal. So that's actually kind of wrong for it to be. If you think of that as a bow leaf, there are a lot of little fungi that do break down the leaf litter and pine needles. But that is not how I would have illustrated it. But this is another picture out of a book that I'll show you in a minute. 3 different fungi on one root. It's another amazing photograph. A lot of the tree species have multiple mushroom partners, and a lot of the plants that we know in the forest have multiple mushroom partners. Now, I like this drawing because it's old, it's British, it's from about 1961, but it breaks down each kind of species of trees with their own little flora of mushrooms that grow mycorrhizally, that might be parasitic, that might be decomposers, and they don't, you know, this fungus you won't find on a pine tree. I think it's pretty interesting, and it's also pointing out how much we don't know of how the forest works as a whole. Maybe some more of these do integrate more than we think, and that's, I think, where a lot of the research is lately. I'm just going to talk briefly here about lichens, in that it is probably one of the oldest associations of a fungus with a living photosynthetic partner, photobiont, green algae or a cyanobacteria. And it turns out that the fungus is the controlling or enslaving partner of the algae to do its work for us. But we see lichens everywhere, especially here in the Adirondacks, this being the lung lichen, which is a good pollution indicator. If the air around here is not healthy, this lichen will not survive. It has to have clean air in order to thrive. Lichens is another whole area, but they're important in the environment of relationships of fungi and green photosynthetic relationships. Okay, just briefly.
It looks like, Susan, I'm just giving you a time check. It looks like we have about 10 more minutes.
Oh yeah, sorry, I'm almost done. I only have about 3 more slides. Sue Van Hook is who you want to invite if you want to know more about this subject. Sue was the mycologist for 10 years at Ecoveda, and this is Gavin McIntyre, one of the co-founders. But Sue used to be at Skidmore as a mycologist and professor there. But Ecoveda is a group that is now growing mycelium into a lot of products. One of the most astounding ones, they're trying to make a substitute for meat, but they're well known for a replacement for styrofoam, growing mycelium into packaging material. Okay, some of us might be home growers of fungi. That's not too difficult. Just have to know a few things about the strains, what kind of logs might work, and contamination will be your biggest problem. Here we have commercial growing of maitake, or the hen of the woods, shiitakes. Oysters will grow on anything. That's probably the easiest thing to grow. I was lucky enough, my club is associated with Phillips. Long story, but anyway. Now my own interest briefly is using mushrooms to dye wool. You can get a lot of different colors out of mushrooms. Here are some of the mushrooms that I collect up here in the Adirondacks. And here are the colors I get out of them. And here's a basket from here in the Adirondacks a few years ago. This group mostly makes the tealy blue-green colors. Now here's some of the books and references that I used for this. These are the two main cultivation catalogs I would recommend. Fungi Perfecti, this is Paul Stamets's bunch, but he's expensive, but if it's in there, he's done it. He's done everything. He's done the work. He does know his stuff, even though I don't think they credit him as a doctor of mycology, but he's expensive. Field and Forest is less expensive, but they are in Wisconsin. They're easy to talk to. This is the book that I've gotten a lot of the pictures that I've used here tonight. Very good photography, little text. And here, Fungi Magazine, I would tell you to get this magazine if you're interested in pursuing amateur mycology at all. It's geared to the amateur, mixed bag of all kinds of articles. And here, this is just a really good book on lichens, although there's a lot of material on lichens. Okay, so here is my end. I'm going to leave this up. And here are some websites, the Field and Forest websites and the cultivation things, Fungi Perfecti, Fungi Magazine, and a couple of really good videos to help. These two down, well, this one here, Suzanne Semard, she's really the one to, sorry, what happened?
We are, looks like, I'm not sure. We can post all of those resources for people on the Grange's website as well, so you won't lose track of them.
Yeah, I'm sorry. I don't know what happened there, but it might have been me. But yeah, so I'm done. So whatever you want to do now, questions or otherwise, or do you want me to?
Great. Thank you so much, Susan. There's so much packed information in there. And I know that it would be so fantastic to do a field outing with you, and we'll hope to try and arrange that sometime in the future. But we do have some great questions that you probably can answer.
Hopefully.
And so let's give them a try. Caleb asked the question, is there solid scientific evidence of the mycorrhizal network being used by trees for communication and sharing of nutrients? Sort of covered that. Any other comments?
Scientific evidence of the
mycorrhizal network being used by trees for communication and sharing of nutrients.
Oh yes, absolutely. Yeah, the one man that I showed, Rita Spigelis at Duke, but there's a number of labs across the country, but it's incredible if you, I don't even know which one of these websites, the Wood Wide Web, probably that top one. There's a lot of, in fact, the one who's done the most work on that actual communication, how trees talk to each other, Suzanne Semard. You should investigate her, because the work she's done with the mycorrhizal fungi is pretty astounding.
Super, that's really interesting. Next question is from Greg, and it says, it sounds like a good understanding of the fungi topic, hence the research interest you mentioned, may help detour the invasive species. Not sure I understand that question, but
invasive species of plants.
There is another question that Greg, if you want to clarify your question in the chat, go ahead and do that. I'll try and refine that. There is another question on invasive species from Mary Nell. Are there specific fungi in the Adirondacks that are considered invasive and pose a threat as a result?
Well, I have to say that invasive species are not always a bad thing. And for some things, what are you going to do about them? They're here, they're well established. They've been here for 70 or 80 years, some of them. I don't think you could get rid of them if you wanted to. And in the case of the ones that the one I mentioned, Swillus bolinus, one thing I did not say is that the mushrooms are partnering with some of these trees, and the trees cannot survive without their mushroom partners. In the case of the Pinus sylvestris, the Scotch pine, I don't think they would have done as well as they have. A planted stand is where they are growing near me, and the mushroom growing with it. They wouldn't survive. The Scotch pine without the mushrooms. Now, granted, there are a lot of other species of mushrooms with those trees. And one of the things that is exciting about fungi is they have an ability over time of jumping hosts. They may have come in with the Scotch pine, but they may be able to jump to a different pine tree, or even in the case of Amanita phalloides. In Europe, it's with oak trees, but here it's with conifers and specifically Norway spruce when I've seen it. So yeah, I can't honestly say that are all invasive species, but you know the problem with fungi, we don't know enough about the fungi to even know what's here, let alone if it's good, bad, or otherwise. Fungi are very understudied. We know all the plants and roses and trees and even the insects we probably have a better handle on, but we do not have a good handle on fungi.
It's that sort of leads to the next comment that the same attendee made. Greg made the comment that he was about ecovative, making building insulation from it. I think the biomimicry is a really, there's a lot of promise in the fungal world for biomimicry applications, and building insulation is one, and the other things that you mentioned, that would be a really interesting presentation to follow up with what we could learn from those things that we don't know that we're just learning about now.
Yeah, Sue Van Hook would be a fabulous speaker for any of this kind of a topic. Not only how the fungi work in the field, but also if you want to know more about what ecovative is doing.
Really interesting.
Yeah, she's really good.
Another question, are the truffles that you presented from the Adirondacks edible?
Oh, well, they're edible to, as Mike Castellano will always say, are all edible to somebody, but not necessarily to humans. He and Jim Trappy work a lot in Australia, and they particularly study the relationship with the marsupials there. A lot of the little marsupial animals depend on the truffles and truffle-like fungi for most of their diet. And the mushroom, the truffles, depend on the little animals to carry their spores somewhere else for germination. So it's a very integral relationship. But yeah, the only truffle we know is the one I showed you here in the Adirondacks. So who is it edible to? The squirrels. That's how Mike told us to look for the truffles, is to look where the squirrels have dug, burrowed into the ground.
Interesting.
That's how we found that one, was squirrels were working on Paul Smith's campus, and we dug into their hole, and boom, there about 6 to 8 inches down was the truffle.
Oh, we have a whole bunch of more interesting questions. We may run out of time, so I just want to let the audience know that if we do run out of time, we will pass your questions on to Susan and we'll check back with you through the Grange. We're glad to get these questions answered, but we do want to try and finish on time this evening. So I'm going to give a couple more questions. Catherine Reinhardt asks, what's your technique for dyeing with the mushrooms? That might be a long one to explain.
It's a variation of natural dyeing. I tend to dry the mushrooms so that I can use them at some other time. In fact, if you can see behind me, all of those boxes are of dried mushrooms that I use for dyeing wool.
Well, you have to talk about your sweater, Susan.
Well, the sweater I have on is also all mushroom dyed. All of the colors are from species of mushrooms. They're very specific species, and the mushrooms have different chemistry, but it's basically crush it up, put it in water, cook it, dissolve out the pigments, and then treat the wool maybe with alum or iron, and then cook them together and see what you get. But
interesting. The first time I saw Susan present at the Adirondack Research Consortium, she was wearing a sweater that she had knit, and all of the wool was dyed with mushrooms, but also all of the wool was labeled by the mushroom that she used to dye it. It was pretty impressive. It's pretty great.
tend to make tags to show the mushrooms that I use.
It's great. Another question is, why are fungi so understudied?
I suspect it's because what we can see is above the ground and only out a few hours to a few days. One of the ones that I'm particularly interested that I stumbled across, it comes out in the end of March, long before most of the spring mushrooms come out. So in studying them, I think maybe 100 years ago where they didn't have food dehydrators, you know, it was difficult to work on them right away, especially if you got 25 or 30 different kinds of fungi, to work on them before they would rot. I'm sure they figured out how to dry some, but still, it's a lot of work to look at. The other problem in this country, and probably in some of Europe, we don't fund our mycology university programs very well. Most of the mycologists that I've sort of known in the last 40 years, when they retire, they are not replaced. So there's only very few really good working labs working on fungi, which is a shame.
Yeah, that is an interesting problem to solve, to also engage young mycologists to try and pressure.
One of the comments in Merlin Sheldrake's book was, we want them to look at yeast, but they want to save the world with fungi. You know, so you have to sort of come to some happy medium of understand the yeast and all of the things in between before you can apply them. That's where these I call them kids, but these fellas at Ecovative, they have really taken the bull by the horns and run with it. They really, it's astounding what they do there. It really is.
Can't wait to look into that more. And it is 8 p.m. I think we are going to, we have tried to have a policy of ending right on time. We do have a few more questions, and I think because the conversation is really interesting, I'm gonna make an exception. I'm gonna break our policy tonight, and I'm gonna throw a few more questions at you if you don't mind, Susan.
Yeah, I don't mind.
And,
but you know, I know if people have to sign off, that's you know, they have to do what they have to do.
And I want to encourage all of you who do need to sign off right now to check back in if you want to catch the end of it on the recording, which will be at the on the Grange's website. So Michelle asks the question, I read in the New York Times that growing mushrooms in apartments is a new fad. Since COVID, it has really taken off.
Yeah.
So grow fungi to eat or to make dyes?
Not so much the ones for dyeing wool. They are still very specific, and some of them are kind of poisonous. In fact, very poisonous. But the edible ones, yeah, the hericium, the lion's mane, and the oysters would be the easiest to grow. Both of these two catalogs that I mentioned, Field and Forest and Fungi Perfecti, they sell kits, and sometimes, I'm not sure if they have ready to go fruiting kits. In other words, you want the bag or whatever you're going to grow in the apartment ready and aggressive enough so that when you open it up according to the instructions, whether you water it or open it to exchange gases, it's going to fruit right away. For the most part, fungi are a little temperamental. They're fruiting, so you know, I can't get them to fruit in my house because it's too dry, especially in the winter. But if you can provide maybe a bag or something over it for moisture, some people have gotten amazing fruitings out of them. I've even heard they're having restaurants grow them as a side thing in their main room, so that people say, I want to eat it, and then they sort of cut it and cook it right in front of them, kind of thing.
Wow.
Yeah.
I'm gonna
be a growing trend.
Let's see. I'm going to go back up to a couple of people who upvoted a couple of these questions at the top. Hey, Tom, nice that you could join us tonight. Tom asked the questions. It says some slime molds can move. Are slime molds a fungus? Are there other fungi that can physically move?
No, fungi, slime molds are not fungi, although they have usually been lumped in with fungi for study. There's a lot of mycologists who study slime molds, but to my knowledge, slime molds are the only ones that are kind of amoeba-like in that they can self-motivate to move on their own. But they are not fungi. If you remember that big pie chart thing in the beginning, they're way off over on the left somewhere, I think. They're not in the kingdom of fungi, literally.
Okay.
Well, like I said, there's a number of mycologists who study them anyway, just because they're so weird and interesting.
Zach has a question. How well do fungi sequester atmospheric carbon? Has that been measured?
Oh, I don't know the answer to that, I'm afraid. But since they are very important in connecting to the roots of the trees, I would suspect that holding the carbon in the ground and being able to turn it back into a useful form for plants is a major component of soil sequestering. Sue would be able to answer that, Sue Van Hook. I'm afraid I'm not clever enough for that.
That's great. We can add whatever references you think of to this list. And we, as I say, we will post this. There's another comment here from a one. It says, I forage. What I found odd is that some regions of my neighborhood, New York, Lake Champlain Beach near Plattsburgh, are basically bereft of mushrooms, a few russula, some lobsters in the fall, and not much else. But then you move half a mile and bang, you have all sorts of boletes, chanterelles, all sorts of things. Can you speak to what geo, soil, climate conditions lead to these stark differences?
Human intervention would be one of them. How built up the area, how disturbed is the habitat, and that might be long range. Whether it's been left alone or whether, if you know the wild center at all, it's basically built on a sand pit. And if you go to the top above the river, you can see a distinct change in the mycological flora right at that point, because the sand pit stops and what was the forest, or the forest after it was logged 100 years ago, is below, going down to the river. And there is many more species of fungi where the forest has been left alone to its own devices. Of the entire community is less disturbed by human intervention. The other thing, the first thing I look at any place when I want to look for fungi, what are the trees? What are the trees that are going to provide the mushrooms that you want? And that's one thing, if you've got a forager, if he's looking for lobsters or for chanterelles, you've got to learn a little bit about what they grow with. They are ectomycorrhizal. They grow in association with the trees. So you have to learn a little bit more about the habitat of the fungus that you want to find. But yeah, human intervention would probably, and that's a polite way of saying, you know, disturbing things that we do to our beaches and our neighborhoods.
Yeah, definitely. It looks like, I think we might have gotten to all of these. One more question here says, do you know much about the nutritional value of mushrooms for humans other than medicinal uses?
Oh yeah, I forgot about that. Yeah, since my interest has always been on identification, they are actually very nutritious. I just never remember what it is that they have. I just don't remember. The Mushroom Institute down in Kennett Square in Pennsylvania has a really good chart about that. But certain little minerals, I think, even some protein.
Anecdotal entirely, but I find that when I eat mushrooms, I have really vivid dreams. And that's not hallucinogenic mushrooms. That's straightforward shiitake mushrooms.
I will say one mushroom that I know, and a mycologist at Phillips told me about this, Tina Eller, is the hericium, or the lion's mane. it actually has been shown to increase, I guess, blood circulation in your brain so that your functioning of your brain works better if you can take or eat some of this mushroom on a regular basis. She takes it herself, which I think is amazing.
Interesting.
Yeah, the lion's mane.
No, go ahead, sorry.
Well, just the lion's mane is the Hericium, of which there are 3 species that grow wild here in the Adirondacks, all of which are edible.
We do find that one in the Champlain Valley.
Yeah, yeah, I would think.
There is a comment here that says nearby natural grow kits are fail safe. I've grown the kits from Field and Forest and others, but nearby naturals are better. So
Yes,
for you home growers, there's a comment for you.
And there's a lot of different companies that do these kits. And I honestly can't speak to that because, you know, it's not been my thing. I mostly get my information from the people who do serious cultivation or cultivation just for themselves.
It looks like we got to all the questions. There are quite a few interesting comments from Greg Pedrick. Thank you for tuning in, Greg. And it looks like he helped the Ecovative get started when he worked at NYSERDA, which is really interesting. So thanks.
Oh, wow. Okay.
It looks like Cordelia threw in a comment that says, check out mushroom-revival.com for a whole range of healthy mushroom tonics. So lots and lots of interesting comments. Okay, I think it is time for us to wrap up for tonight. I just really, really want to thank you again, Susan, for your great program, and we look forward to having more questions answered later. If people have an interest in additional questions, let us know. We'll do our best. And we appreciate you sticking around with us for the end of the program. And so we look forward to seeing you. If there's any questions we can answer, contact us. And if you haven't already, sign up for the next Lyceum presentation. And again, thanks for coming tonight.
Thank you. This has been fun. Real challenge for me. I had to learn a lot just to be able to explain some of this myself.